Micheletti. I'm a professor in biochemical engineering, University College London, and co-director of one of the two UKVN-funded hubs called Vax-Hub. I'd just like to start to say a few words on how the idea behind the event came about, and acknowledge my friend and colleague Gilbert, who later last year proposed a lessons learned event at the back of one of the vaccine CPD courses we run in UCL. Me and Professor Sarah Gilbert, the co-director of the Vax-Hub, really liked the idea. And this gave us also the opportunity to work together with the Imperial Hub team and really further develop the original idea into today's event. We certainly didn't expect so many registrations, participants from very different sectors, from so many countries. Apologies to our colleagues in North and South America. We know the choice of timing is not ideal, but we hope we can enjoy the event once the recording is made available. The popularity of this event certainly shows that there is interest in exploring some of the questions we have set for reflecting on the role of academia, on the importance of collaborations, on discussions on global approaches, and I really hope you enjoy what we have put together. So I'm going to hand over to Ben for the introduction to the event.
Thank you, Martina. So I'd like to offer you a welcome from my side. My name is Ben Pierce. I am Operations Manager of the Future Vaccine Manufacturing Research Hub from Imperial College London. It's been my pleasure to co-organize this event with Vax-Hub, Martina and her team. It's been a delight to have so many prestigious speakers and panelists. And we'd like to give our gratitude to you, the attendees of today's event. And where we'll be focusing on vaccine manufacturing during a global pandemic, what we have learned from COVID-19. But I do want to begin with some housekeeping notes. And just before we get to the session one, race against the clock. Please be advised that the event is being recorded. You have been notified of this beforehand, but the recording began just a few minutes ago. We encourage everyone to enter any questions in the Q&A section at the bottom of your Zoom menu. For the Q&A sessions at the end of each section, we have early career researchers from our hubs as co-chairs who will be selecting and collating questions that will be presented to the speakers and panelists from that session. We will also be presenting some poll questions throughout. Please do let us know your feedback. We're very interested to hearing what your responses to our few poll questions will be. And we would also like to offer our great thanks to our funders, the Department of Health and Social Care. And we have these hubs set up through UK aid funding that's being managed by the EPSRC, a research council within the UK, now part of the UK RI. We would like to point out that the views and opinions that you may hear today are not necessarily those of the Department of Health and Social Care and our funders. But we do encourage you to highlight the event on social media. We've created a hashtag. Please use the hashtag shown at the bottom of this screen to circulate interesting points and outcomes, questions, thoughts, opinions about the event. Very, very much looking forward to seeing it being circulated from today. Now, turning back to the Department of Health and Social Care, I would like to point out how the hubs have been funded and how they began. From 2016 to 2021, the UK government made the decision to commit £120 million of UKA funds to support research for vaccine manufacturing, vaccine development against pathogens of particular importance in low and middle income countries or developing countries. And a stream of research activities was generated through that under the guidance of the UK Vaccine Network, or the UKVN. You may see that acronym throughout today. And from that, two innovative future manufacturing research hubs were born in 2017, but really began from 2018. And just to avoid any confusion, One is called Vax-Hub, and that's from the University College London, co-directed by Professor Martina Micheletti and Professor Sarah Gilbert. And the hub where I'm a part of, as operations manager, is called the Future Vaccine Manufacturing Research Hub, but for brevity for today, I will be calling it the Imperial Hub throughout. That's, of course, from Imperial College London and directed by Professor Robin Shattock. So these are the two hubs that will form the consolidated summaries of activities that you'll be presented with today. And just note that it is just under 17 million pounds and a relatively small portion of a vast amount of research that has been allocated from this 120 million pounds through the UK VN stream. This is not an exhaustive list of all the projects. There are some others, but just note that with the announcement from November 2020 that the UK is cutting UK aid from 0.7 to 0.5%. That's a small number in terms of percentage, but it amounts to nearly 4 billion pounds. We may see severe impacts and more barriers to our engagements and support for vaccine research, not only globally and particularly in developing countries, but also with severe impacts likely in the UK as well. So with that brief introduction, I'd like to hand it back to my colleague, Martina. Off to you, Martina.
Can we have a next slide, please? Thank you. It is my pleasure to present the agenda today. We will start with reflecting on the race to produce COVID-19 vaccines and really on the research behind these efforts with some of the UK key players. Ben will then be your chair for the next two sessions, which will focus on the hub's role in supporting global manufacturing capacity building. We will then move to discuss pandemic preparedness, and then in the end, the hub directors will draw some conclusions on the lessons learned and highlight what should be the focus in the future. Just a reminder, questions are going to be handled after the last speaker for each session. So please do use the Q&A chat. And if you wish, please name the speaker the question should be addressed to. We have also set a few polls to engage with the audience. They're going to show you on your screen, so please answer if you wish using the poll function. I think the first one is our icebreaker and is shown or should be shown to your screen shortly. I'm now going to present our first speaker. It's my pleasure to introduce our keynote speaker, Professor. So Steve Bagshaw, he has over 35 experience across the biotechnologies and fine chemicals business and manufacturing sectors. He was CEO for 6 years of Fujifilm Biosynth Biotechnologies. Steve chaired the Importers Board, the UK Industrial Biotechnology Leadership Forum, also co-chair of the UK Bioeconomy Strategy Board. Is currently working with the UK Vaccine Taskforce as an industry advisor, is non-executive director of CPI, and has been a council member of one of our councils, the Biotechnology and Biological Sciences Research Council. His efforts were recognized by many awards. I'm going to mention one which is particularly dear to me, which is the Peter Daniel Award for contribution to UK bioprocessing. Over to you, Steve.
Great introduction, thanks, and dear to me too, that award. That's lovely that you should say it like that. I got a chance to look back at the way in which the Vax-Hubs were set up back in 2017, and as I read the stories that were written back then and the applications, what struck me was that it was talking to me from a land that was like a foreign land almost in terms of where 2017 was compared to where we are now. In 2017, of course, pandemic was something that we were talking about as lessons learned from SARS, from swine flu, from MERS and Ebola. Whereas today, we're all significantly wiser in what it means to deal with pandemic, with global scale manufacturing, and the need for effective vaccines. What I'm hoping is that in this event, you'll get a chance to look back, but also you'll use the lessons learned in a way in which we've done so far from 2017 to today, but accelerating them so that the next 15 months of pandemic is informed in a way in which the first 15 months only started to be informed. You can see that as you start to get into this, and you see society changing on our watch. Martina talked about the years in which I've been working in biopharmaceuticals. And throughout that period, whenever I tried to explain to somebody what exactly I was doing, you could see their face glaze over as you got to biopharmaceuticals. You didn't even have to start talking about manufacturing. It was just enough, the name. If you added advanced therapies or even got to vaccines, no one really was still listening. Now what I've found is a complete difference. People understand that if you're working on vaccines, it's important. And last year, as those of you who are following me will know better than I do, everybody was interested in what happens next. The question on everyone's lips was, will we have a successful vaccine? Will we be able to create something that's safe? Will it come by Christmas? Would you take it? These were the questions that many of us were getting asked. No matter how small or big the role was you were playing in vaccines, if you said you were working on the vaccine, then you were subjected to questions and answers. What I find now as I walk around is that when people ask me the question, what do you do? And I say, well, I work with the Vaccine Taskforce. Almost immediately their answer is, well, thank you. Because there is such a change in the way in which people now perceive the role of vaccines and in the way in which normal life, whatever that means, can come in the future, and how dependent we are as a globe on successful deployment of vaccines. I was embarrassed the first time someone said thank you to me, because I realized what a small role I was playing in the story. But what I realized as more and more people said it to me was that I needed to pass that on. And so I pass it on to you, the thanks of my neighbors. My neighbor who texted me two nights ago to tell me that her husband and she had just had a second dose, and she said thank you. And so I pass on her thanks too. And I know that there are people around the world who would join me in saying thank you to all of you who played some part in the development of vaccines to deal with COVID-19. For many of you who would describe yourselves as scientists, you've also seen another change, and that is that society sees scientists as trusted voices in this pandemic. Some of you who've been exposed to the press may not think that, but as a whole, the scientific community is seen in a better light now than it was, and is one of the trusted voices of what happens next. What should we be doing? What does the science say? Who would have believed that there would be headlines like the Deputy Chief Medical Officer having to turn down Strictly Come Dancing? It just wasn't something that anybody thought of 12, 15 months ago. And yet his has been one of the trusted voices throughout this pandemic, and someone that people listen to. And so for many of you who've been on the radio and who've been on television, I also say a huge thank you for putting yourselves through that and explaining to people just how it is from your aspect and how you see the science. Because not only are you explaining it to people, you're also inspiring a generation of people who are thinking now that science matters and science is cool. And that means that young adults, children, and particularly parents who are helping their children make decisions are seeing the option of taking on science as being something really important. And in the long term, that's a fantastic thing for us all. The issue with COVID-19, whenever we come and talk about it, is that the stories that we tell often start to get personal too. So all of us will know someone who knows someone who's been affected by COVID-19, a life lost, someone who this time last year was with us and who isn't with us today. And that's still going on all around the world. And you see that in your pictures on the news. And anybody who says that this crisis is nearly over is not really looking beyond the end of their nose. There is so much still to do in this area. And so all of what we're going to talk about today matters, and matters immensely. in a way that back in 2017, many of you who signed those papers knew it would do one day, and it does now really matter to the world. So let's talk about the response of how we responded in this country to the COVID-19 crisis. And I'm being followed by two eminent speakers on this, so I'm not going to spend too much talking about the way in which Oxford and Imperial worked. I'm going to talk more in general about how we as a community responded. The government appointed a task force at the highest level, responding to the Prime Minister back in April last year, and that task force brought together experts from industry, from academia, and from government, and looked at ways in which they could somehow plot an end-to-end journey to get to mass vaccination in this country. Given three targets. One was to get access to the most promising vaccines for the UK population. Second, to make sure that they were available internationally. And thirdly, to put legacy in place such that we were better prepared for the next pandemic. In bringing together the academics who were working on that, we realized that in hindsight now, that there were decades of experience being sought and used and worked with in that collaboration, both as individuals and institutions. And that through the period of the past 5, 10, 15, 20 years, that had also been backed by, although you may not feel so when you're in the funding rounds, a consistent level of investment, such that institutions and individuals were able to have careers and institutions were able to grow in terms of the way in which we address vaccines and vaccine development. Dame Otterline described it as being a flexible, agile, empowered, interconnected network of innovators, scientists, and researchers. And that when you bring this together in the UK, you have something special, something that is very much a national treasure, and something that needs to be invested in. And I'm sure as we get to the end of this and look to the future of where initiatives like this go, that will be another piece that we come back to. And yet in a pandemic crisis, that isn't enough. It isn't enough to have something that has as deep an expertise as that. It has to be joined up, because end-to-end acceleration requires us to break down barriers in all sorts of directions. And so what the Vaccine Taskforce did was to bring together expertise, not just from academia, but also from industry and also from government. And it sponsored conversations and the chemistry between individuals. You'll have read in the press of CEOs who met researchers and recognized there and then that they needed to act, and were able to bring the might of an industrial powerhouse behind certain academic institutions or ways of working. And though we may look back and think of some of that as serendipitous, we will also see that around the world, governments, academia, and industry realized that they were better together than they were apart, and that process acceleration would only come when those relationships started to also be turned into some kind of process. And so the Vaccine Taskforce spent significant time through the summer of last year doing due diligence on hundreds of promising vaccines, getting to a list of multimodal vaccines and a recommendation of which were the most promising and which were the ones that should have deals done with them. And you know this across adenovirus, across the mRNA, across the inactivated virus, across protein subunits, across plant-based, across some of the antibodies, some of the treatments that are out there, all of those being assessed and all of those being backed then by either a deal or investment. Rapid scale-up, process establishment, qualification and approval was all happening in parallel with that due diligence in the different supply chains associated with each vaccine. And what this government did, and the Vaccine Taskforce recommended, was that capacity was reserved, capacity was invested in. So you had Oxford Biomedica, Wockhardt, Cobra, Valneva, all invested in. Then you have Fujifilm Diosynth, GSK and CPI also, with capacity reserved and investments made. Facilities that had been commissioned, like VMIC, were re-energized or not even re-energized, just energized and made to come quicker, but with a much bigger scope. As were some of the investments made at Porton Down and some of the testing facilities. and across the network, and a repurposing of assets such as the one at CGMIC have now. That's the Cell and Gene Therapy Catapult at Braintree, where large-scale commercialization can be done of vaccines that no capacity was available to be done in the UK, and now can be done in Braintree. Alongside that, skilled people were secured, and supply chains that, known to be global, backwards and forwards up the line, were looked at, and partnerships, relationships made to try and secure all that was needed to make sure that you could scale up successfully anything that was a successful vaccine. In the early days, we like to think of not dropping the ball. We were putting around people a set of supply chain experts who would just listen and say, so you've got a problem with bags, let us sort that out. You've got a problem with that solution or that test, let us try and sort that out. That was the kind of support that was happening in the early days. And I'm delighted to be sharing the platform today with two people who were carrying the ball at that point, and we were in the Vaccine Taskforce trying to support them carrying that ball. But as well as making sure you make population scale doses, you have to have them approved. And so the clinical trials processes were also being revamped and looked at. And so a registry was put together with 500,000 people volunteering to be involved in clinical trials going forward, to try and shortcut the time it would take to set clinical trials up. Similarly, in terms of the regulator deploying rolling submission of data, parallel analysis, and getting to the end, so that if you think of that project plan that traditionally goes like this, with another item and then another item and then another item, and the date just keeps going right across the page. In this, the date was drawn left across the page, and the regulator was able to, within days of being given the last piece of data, have an opinion on how approvable the product was. These are things that we'll look back on as ways in which we transformed the way in which you could get clinical trials done and get to approvals. But even now, if you approve a vaccine, you've still got to deploy it. And so in parallel with all of this, starting in the summer of last year, was the NHS efforts to make sure that we had the largest mass vaccination rollout that we've seen in this country. And so the NHS, working with their own professionals, but then across the nation, and all of you will have enacted in the UK, will have encountered somebody somewhere who's volunteered, who's played a part in either vaccinating or being one of the welcomers or running the phones or doing some part of that last mile of getting the vaccine from the warehouse into the arm of the people. And so all of that's happened in parallel with a huge civil service activity to remove roadblocks and to get funding. And so that trio of government, academia and industry has been a real success story of how you bring civil servants alongside academics and industry, working within the funding mechanisms that there are and creating new funding mechanisms where they were needed to actually build the infrastructure that was needed to roll out the vaccine. So where we're sat today is that we've got 2 billion doses out globally. We've got 69 million doses in the UK that have been given out. It's not over. It's not enough. We need to keep going down that track. However, pause for a second and say, a year ago, would you have predicted 2 billion doses by June the 10th next year? That's as long as you get in the vaccine world, though. That's the pause. You go, yes, great work, but look at how much more there is to do. So let's just quickly finish then by talking about the things that we've got going forward. Continuing manufacturing and deployment, promising vaccines in development. There are still vaccines that we need to get to market. We need to get them because they're the ones that will help us get global scale production. There are partnerships to deal with variant that we're undertaking. We're looking at more investment in sequencing, in testing, in clinical trials to help us, and embedding the learning from the last 15 months. And looking forward, we need a deeper and more vibrant, a more resilient vaccine ecosystem to be prepared and more ready for next time. And that ecosystem is being developed by working with developers, bringing in larger scale looking at the larger scale manufacturers and looking at what would it take to have vaccine manufacture here in the UK. We're looking at supply chains and how we can bring those onshore. We're looking at technologies in terms of innovation and research around manufacturing, formulation, and clinical studies, and looking at the ways in which we can improve deployment, and very much taking some of the echoes of the Vax-Hub work, and across government, making sure this is joined up. Too often, many of you will have been subjected to the fact that there are people who work with you on one line, but no one knows what the left hand's doing or the right hand's doing. And so there's a real piece of work being done to try and join up our response. To me, this is the tribute to the past 15 months that we're paying here, and to the past 3.5 years of the Vax-Hubs. It's a positive legacy that comes out of the work that was done by yourselves and what's happened in the last 15 months. It will help us with the next 15 months, but more importantly, it's also got to keep us ready for the next one. Because one thing 2017 to 2021 has taught us is that we never know when the next crisis is going to come, but it's likely to be sooner than we think. And with that, I say thank you.
Thank you, Steve. Thank you for highlighting all the work of the Vaccine Taskforce, but especially the role of science in the society and the impact on the next generation. I think we're going to explore some of the points you made in the Q&A session later. So for all attendees, if you have questions for Steve, please post them in the QA chat and we'll discuss with Steve later on this morning. So I'm going to now introduce our next speaker. It's a pleasure to welcome Professor Robin Shattock. He's the head of mucosal infection and immunity within the Department of Medicine at Imperial College London. His research focuses on the mechanisms of mucosal infection and the development of novel preventative strategies appropriate to a developing world setting. So he has secured funding from very different sources. I was going through all his projects, many interesting ones. Gates, EU, Wellcome, NIH, MRC are some of the funders, and he's the director of the Future Vaccine Manufacturing Research Hub at Imperial. Over to you, Robin.
Thank you very much, and good morning, everybody. So welcome this morning. I'm going to be talking to you about one aspect of the Future Vaccine Manufacturing Research Hub that we have been focusing on, and really started working on this back in 2017 with the view that pandemic threats was something that should be taken seriously, and we needed to have new technology to prepare for such an eventuality. I'm not quite sure. Oh, there we go. And so we were already looking at RNA technology for a number of reasons, partly because it allows you to manufacture on a very low footprint with small capex. It allows you to do rapid production and to make potentially millions of doses within weeks, and meets that just-in-time response capability for pandemic X. And we've kind of put this forward as an approach, a new approach for pandemics at the World Economic Forum in 2018. And I think we had about 30 people turn up to listen to the talk, and they probably got lost and just turned up off spec rather than deliberately searching us out. Because at that stage, people were saying, well, you've talked about pandemic threats, and people have been mentioning this for the last 10 years, but it never seems to quite happen. Of course, hindsight is a beautiful thing. But one of the reasons that we were very interested in this technology is really this small footprint approach. And we had the vision, and still have the vision, that you could set up manufacturing sites in the equivalent space of 2 shipping containers. And it would be paradigm breaking in that rather than having to have large warehouse-sized manufacturing facilities that supply the world, you could have network facilities around the world ready to build RNA, And then the design of the vaccine becomes more like the software. So in the same way that the personal computer allowed access to information and the internet, we think that in the future, small-scale manufacturing may allow global access to be more readily built for future pandemics. And certainly one of the things that we're seeing as a result of the COVID-19 pandemic is an explosion in countries interested in wanting to set up mRNA manufacture in different parts of the world. So this is definitely the way the world is going. And we were really starting to develop this from the beginning. Well, just to give you a quick tour of the two types of RNA vaccines that are out there at the moment. There is conventional mRNA vaccines, and you'll have heard of these because these were very successful, both from Moderna and Pfizer. Getting emergency authorization, and millions of doses of these vaccines have now been administered. These are relatively simple. They encode a gene of interest, and in this context, it was the glycoprotein of SARS-CoV-2. They need to be encapsulated in a lipid nanoparticle to gain entry into the cells, and when they're in the cells, they carry the code for the spike glycoprotein and engage the cellular machinery to make that protein. So if you like, it's moving away from manufacturing a complicated vaccine, either recombinant proteins or virus, and really using the cell's machinery to make the vaccine itself. Well, we have been interested in self-amplifying RNA, and the reason for this is because it allows you to use lower doses. Self-amplifying RNA is based on, typically on alpha virus backbone, where we use the non-structural proteins that encode the polymerase that allows the RNA to be amplified once it gets into the cell. Now, it has commonalities. It also needs to be encapsulated to deliver it into the cell. But once in the cell, its polymerase machinery makes multiple copies of the RNA. And so essentially you get more protein out with a lower input of RNA. We think this is going to be important in the future because you can look in terms of the doses of RNA that's required for Moderna, 100 micrograms per dose. Pfizer-BioNTech is at about 30. And we are targeting a dose of 1 to 5. We still our ideal is to get down to 1. This is important on a number of issues. Firstly, it allows you obviously to manufacture much more doses for the same volume of manufacturing material. It also means that the safety profile is better because there is some side effects with higher doses of RNA. But perhaps more critically, it also allows us to think about combination vaccines, where if you were looking at a combination of, say, influenza, 3 influenza targets and a COVID-19 target, you'd be looking at a 4 microgram dose as opposed to a 400 microgram dose. So we also see this as very much technology of the future for multi-component vaccines in the RNA space. Well, we started our journey in January of 2020 by engineering spike glycoprotein. And like many, we used data from the literature. We were able to make a prefusion stabilized version of this. We thought this was important to maximize the induction of neutralizing antibodies. And we were able to move with support from the Vax-Hub, the Vaccine Hub, in terms of funding into preclinical models. We saw that this vaccine was very, very potent in small animal studies. And then more recently, and perhaps Sarah may touch on this in her talk as well, we were able to see, again, in small animal studies, that it's very complementary to other approaches, and it can either prime or boost viral vectors. And this may be important in terms of thinking about how these technologies are used in the future, where one can mix and match different vaccine candidates. And there are some obvious advantages there, both in terms of enhancing immune response, but also availability of vaccine doses. Well, we were working on the manufacturing process in our hub at the beginning of the epidemic. And really at this time, it was very much laboratory scale, looking at the work to start to do process innovation and scale up when suddenly SARS-CoV-2 appeared on the horizon. And so we had to work out very quickly how to manufacture a vaccine, having never done this ourselves from an academic background, rather than coming from it from a well-formed commercial entity. And what we had to do was work out how to go from sequence into production process to rapidly produce clinical quality grade RNA and to formulate it. Now, to do that, we had to make some very quick judgments and decisions. And so we had to take off the shelf a formulation that had been developed for messenger RNA. In fact, it's almost an identical formulation to the one used by Pfizer-BioNTech. And so we were taking a technology that had never been into humans before. So a very different approach to any of the other vaccines that you've heard of that are out there. We were going from sequence with a new technology to first in human studies in a record-breaking time. Now, of course, we didn't have the advantage that other RNA companies had, that they had 10 years worth of development time and several clinical trials before they embarked on this journey. And so in some ways, perhaps we were really pushing the envelope very fast in order to get an early clinical signal. Now, in the context of the pandemic, you have to remember that when we started this journey in January, this was not a pandemic. It was still an isolated outbreak of a new virus in China. And we were thinking at this stage, well, we might be developing a vaccine that, if it was successful, might be used in China, rather anticipating that we would be looking at it in the context of a global pandemic. And of course, the attractiveness of the approach is that we could potentially produce within a very small reaction volume millions of doses, and that's still something that is realizable. Well, what we were able to do is to move from that sequence. In fact, we didn't start designing a vaccine until the 19th of January. We'd sat on the fence. Seems strange now to think about that. And we were able to assemble in very quick time a vaccine and get it into preclinical animals. Again, this was really only achievable because we had funding through the hub system, and that allowed us to get into a preclinical toxicology study without having to wait for grant funding from peer review mechanisms. And in fact, one of the lessons we learned was that the peer review process really was very slow, and if we'd relied on that exclusively, we wouldn't have made the timelines that we were able to realize. Now, because we weren't a company, we had to very rapidly look for CMOs to manufacture our vaccine. In fact, we had to turn to a company in San Diego, TriLink, to manufacture the RNA, and another company in Austria, Polymune, to formulate the material, working with a Canadian company on the formulation, to supply the formulation for that process in Polymune. So again, in terms of looking to the future, we definitely need to build that capacity within the UK to allow this technology to move much faster. But we were able to get into the clinic by the middle of June with a technology that had never been in human studies before. And I think that that was probably not the fastest in terms of vaccine development globally, but the fastest that anybody has ever done that with a brand new technology. Well, the rest is history, as it were. We went very quickly into dose escalation studies. Remember, we had no idea about the potency of this vaccine in humans, so we had no reference, unlike the other technologies that had a reference of previous clinical trials. We went through dose expansion and a large safety trial involving over 400 participants. And we were planning to move into efficacy studies this year, but events really overtook us because of the successes of the other companies. Now, that's a fantastic outcome. You have to remember, again, early in last year, people really didn't know whether any of these vaccines would work. And so it was important to have as many strategies moving forward in case only a few came through with a successful outcome. But in fact, the really great news is that many vaccines came through with a very high bar of efficacy, particularly the work from Moderna and BioNTech. I won't talk about the adenoviral vectors because Sarah will cover that very nicely in the following talk. So our vaccine was shown to be safe and immunogenic. I can't show you the data. We've submitted those for publication, and we hope that that will appear on a preprint server literally in days. However, we took the view at the time that when we got the data that it wasn't as good as those vaccines that were already out there with our first generation approach. And so we have been re-engineering, redeveloping and improving on the vaccine, and we have other trials, which I'll talk about in a moment, that are moving forward. Of course, other people's success have made it more problematic for us to move our vaccine further forward in terms of it makes those efficacy trials more difficult. And so, for example, we've started a new vaccine study, and it's becoming quite complicated to find people in the UK who've not already received vaccination, who want to receive an untried novel vaccine approach. We also recognize very much that, in fact, we lacked pharma backing, and that was something that really set us back, perhaps, in terms of moving forward. And so we've been working to change that. And perhaps in the questions we can come back to why pharma was less willing to take a bet on this new technology. However, we see that that's changing. So in terms of what we were able to do, we were able to advance this approach by probably 5 years. We now have variant vaccines that we have developed. We're looking at potential boosting strategies with different funders, and we are focusing technology very much on other indications, because although this has been very much about COVID-19, the technology is very much a platform technology. We were very much welcome the support from MRC UK, from NIHR and the Vaccine Taskforce to get into clinical trials. But again, what we noticed is, in fact, we would have not been able to do the job without philanthropic donations. And this was really probably matched to the government support that we got. And this allowed us to progress without that stop-start scenario that you get with conventional grant funding approaches. So for the future, in terms of pandemic response, perhaps different funding mechanisms need to be considered. And one of the things we recognized very quickly looking back is that, in fact, we were really able to keep up into this early part of the clinical trial program. But once it moved beyond that, we were really falling behind based on investment. And you can see that the large levels of investment that the RNA companies got in order to get through efficacy and be first to market, compared to the investment that was provided for the Imperial program. That's not really to complain, but what I think it highlights is that the companies in Germany and the US that were able to respond in the RNA space actually had very strategic investment from their governments, both during COVID-19, but also in the preceding 10 years. And I think if we want in the UK to be an important player in the RNA space, there are some significant lessons we can learn from these other countries in terms of their investment in innovation and manufacturing in the RNA space. Well, one of the things we recognize very quickly is that RNA is really at the start of its journey. And one of the strengths, but also defects, is that when RNA enters into the cell, it's recognized as, by the innate immune system. This provides an adjuvant property, but it also serves to reduce the expression of RNA, because part of that antiviral approach is to reduce expression of RNA within cells that have been alerted to RNA being introduced into the cytoplasm. So another approach that we were able to develop with funding through the Hub and also funding for Anna Blackney through a Marie Curie Fellowship, is that we were able to start to look to the future. And this represents work where we looked at inserting small components of other viruses that actually dampen that innate recognition and switch off the antiviral pathway, and encoding that into our vector. What we identified in this initial screen were two small proteins, ORF4A, interestingly from the MERS coronavirus, that actually shields double-stranded RNA from being recognized by some of the innate pathways that trigger an interferon response. And using funding from Innovate UK, we've now taken this into a new clinical trial that's just started this month. And we're hoping with funding through our hub to start a trial in Uganda. That's dependent on us getting a no-cost extension. But if we're able to realize that, that will be the first study of a self-amplifying RNA vaccine in the African continent. And so we're keen to be able to do that, both to demonstrate safety and look at response to such a vaccine in a different population. The other thing that we learned through this journey is that the way you present the RNA is particularly important. I mentioned that we had to take an off-the-shelf formulation. It hadn't been optimized for self-amplifying RNA, which is different to mRNA. And what we do have in the UK is a wealth of expertise, not just within our hub, but wider in academia, in formulation, in lipid nanoparticle approaches. But at the moment, it's very fragmented and not coordinated. And so a way of harnessing that approach for the future will accelerate UK's competency in this arena. Some of the things that we've been focusing in our hub are ways to move from ultra low cold storage of RNA vaccines through to storing at 2 to 8 for over 6 months and to room temperature for months, particularly work in our hub that's been led by Ranjin Chen and Jason Hallett, but also working with others outside of the hub. And so we think that very readily in the future, although RNA technology was limited for the pandemic by the cold chain, we will be able to solve that issue moving forward. The other thing that we were...
Hey, Robin. Sorry to interrupt. This is all very interesting, but we have a lot of questions and I want you and the others to have an opportunity to answer. So if you could...
I will wrap up.
Thanks a lot.
Very close to the end. So we also have been investing in manufacturing through CPI, partnering through the hub, and then picked up by the Vaccine Taskforce and quality control, a very important aspect of RNA vaccines that still needs to be resolved. And so we've been developing a pipeline of RNA, and I think the RNA approach is really very open to this. We have clinical trials funded by Innovate UK, funded by the CEPI program, and other partners moving forward into the future. And we think we're really very much at the beginning of this journey because RNA technology not only can be applied to infectious disease vaccines, oncology, but also a whole host of other indications. And so we've set up a spin-out company in order to support that. We're looking hopefully for other commercial partnerships. But I want to finally end on really we need to learn the lessons from other parts of the world that actually if we want to be players in RNA technology of tomorrow, we need to make ensuring that there is investment and forward thinking to make sure that it's available and invested in today. And I'll thank you there.
Thank you, Robin. And yeah, thank you for sharing the journey, but also highlight some of the challenges and some of the needs in terms of wider UK manufacturing capacity. It's a pleasure to introduce now Professor Sarah Gilbert. So she's a Vax-Hub co-director from University of Oxford, a professor of vaccinology, co-founder of the company Vaxitech. Her research focuses on development and testing of new vaccines designed to induce strong and protective T and B cell responses against many different viral pathogens. She has worked on influenza, malaria, Nipah, MERS, Lassa, among others. And as you probably all know, she led the efforts that brought us the COVID-19 vaccine in collaboration with AstraZeneca. Over to you, Sarah.
Thank you very much, Martina. pleasure to follow on from Robin. Presentations from each of the hubs showing what we've contributed to the pandemic, which I think you'll agree has been quite substantial, and it was very good foresight to have these hubs working in different technologies, but both providing very useful outputs. So I want to take you back to the Ebola outbreak in West Africa in 2014 going into 2015, because this was a catalyst for many people to realize that we really should be doing more to develop vaccines against pathogens that we already know about that can cause outbreaks. Now, Ebola had previously been identified, I think it was 1976. It had caused a number of small outbreaks. Somewhere in West or Central Africa, there's probably an outbreak every year, often in very rural, isolated areas, contained by contact tracing and quarantine, and there hadn't really been a great deal of effort to develop a vaccine against it. But what was different in 2014 was that it got into densely populated areas, into the cities where there was more opportunity for the virus to transmit between people. It spread between countries and rapidly developed into a much larger outbreak than ever been seen before. And at that point, we really needed a vaccine, and there wasn't one available. Now, some work had been done mainly in the US by a defense program to develop vaccines against Ebola to be used as a bioterrorist weapon potentially, but not really thinking about rollout in countries that might experience outbreaks. And so there was a lot of work to quickly bring on that vaccine development and to test vaccines in West Africa to see if we could then have a vaccine to deploy to end this outbreak. And although there were 3 countries mainly involved, Liberia, Sierra Leone and Guinea, there was spreading to other countries, small number of cases in other countries, starting to see some international transfer as well. This was a much more slow burning outbreak than the respiratory pathogen SARS-CoV-2, but it did have the potential to spread much more widely. And so it was recognized that something needed to be done about this. I was involved in the early trials of one of the vaccines that had previously been tested in non-human primates and shown to be effective. It wasn't quite ready to go into clinical studies. We accelerated that. We got the vaccine put into vials, ready to initiate clinical studies. We did those in Oxford. We transferred data and vaccine to Mali, one of the neighbouring countries, to the outbreak area. A further trial was done there. And we then expected that to go into an efficacy study to see if the vaccine actually worked. We knew that the reactogenicity was acceptable and that immune responses were being generated, but would it protect people against Ebola in the way it protected the macaques that had been immunized and then deliberately exposed to Ebola? What happened was that there was quite a long delay between getting that early safety and immunogenicity data and initiating an efficacy trial in West Africa. And while the plans for the efficacy trial were being discussed and the ethical way to do this trial was being debated, the incidence of cases was declining. And if you're going to test vaccine efficacy, you do have to have cases in which to determine that the vaccine's actually working against. And by the time the efficacy trials were ready to start, only one vaccine actually was tested and went through that process, and that was Merck's VSV vector vaccine expressing the Ebola glycoprotein. Fortunately, it had extremely high efficacy, and it was then able to be rolled out at the very end of the outbreak, although in reality, the outbreak had really been contained by contact tracing and quarantine, and the vaccine was just useful for mopping up a few small hotspots at the end. The difficulty of using this vaccine was that it requires ultra low temperature storage, minus 80 degrees, and the other vaccine that we'd been involved in testing, the GSK's Chad3 vectored Ebola vaccine, didn't require ultra low temperature storage. It's 4 degrees. It has a much more mature manufacturing process, could be made in very large numbers of doses, but it didn't get into an efficacy trial, and so therefore wasn't licensed for emergency use because there wasn't proof that it would actually protect against Ebola. So the Ebola outbreak and the control of it prompted a lot of looking back and lessons learned, as we're doing now, following the SARS-CoV-2 pandemic. And the WHO set out their R&D blueprint, encouraging vaccine developers to develop vaccines against the known pathogens. And the technology that we wanted to use for this was our Shattock-1 replication deficient adenoviral vector vaccine, which is another platform technology, very adaptable, you have the technology that you do a lot of development work on, understand the manufacturing, the testing, the dose to use in humans, the reactogenicity, the immune response that's generated. And with all of that data on the platform, you can then very rapidly go into producing a vaccine against a number of different diseases. Now, for outbreak pathogens, where there's not any market to make money out of selling these vaccines, there may just be stockpiles, there may not be widespread use, but we have to be ready to use them if there is a large outbreak and try and prevent that outbreak from spreading more widely and potentially becoming a pandemic. It's very attractive to have a platform technology because a lot of the costs are associated with the development of the platform rather than the development of the individual vaccines. So once we've done the development on the platform, we have to spend less time and, importantly, less money on development of any of the individual vaccines. We do, of course, still need the clinical trials. We need to show safety, immunogenicity, and efficacy of all of those different vaccines, but the huge amount of work that's required on manufacturing, quality control, logistics of distribution of the vaccine, that applies to the platform, and it's already been done. So it's a replication deficient simian adenoviral vector vaccine. That means it doesn't spread through the body. It doesn't get attacked by any antibodies that people have already got through exposure to human adenoviruses, which is a common thing to happen. And we'd already got quite a lot of work done on the use of this vaccine in clinical trials, including, importantly, in a trial of a vaccine against another coronavirus, Middle East respiratory syndrome. So we took the approach in the Jenner Institute of using this technology widely and starting a whole list of programs. You can see the different diseases on the left to make the vaccines and work through what we needed to do to get them tested. So we've been doing this in 2019 and continually adding to this table. And the MERS vaccine development was highly relevant to the SARS-CoV-2 pandemic. Just a bit of background on MERS. Most cases occur in Saudi Arabia. It's endemic in camels in that population. It can spread from camels to people. They have outbreaks in hospitals from time to time. And there was one very large outbreak in South Korea, which involved 4 hospitals. It's only fatal in people who are older or immunocompromised, but there are infections in younger people, usually asymptomatic infections. So the fatality rate is probably overestimated because all of those mild or asymptomatic infections are not being detected. But the plan for this vaccine was to have a vaccine that could be used as a stockpile in case of a large outbreak, to vaccinate the healthcare workers in the hospital who themselves can become infected and then be an important means of transmitting the virus to other patients. A vaccine for travelers, a vaccine for people who work with camels, and also the idea of vaccinating the camels, because this virus has only been present in camels in the Middle East for about the last 50 years or so, and the incidence there has increased in the camel population. If that could again be reduced, then the problem of MERS in the Middle East would go away. And we had completed a phase 1 trial of the vaccine in the UK and had one underway in Saudi Arabia. So we then drew on our experience of the MERS vaccine and the development of the Shattock platform to make our COVID vaccine, again, the spike protein from the coronavirus put into the Shattock. We did our first manufacture in Oxford, and these are the vials of vaccine that we made and had available by April to go into our first clinical trial. We took blood samples, we did lots of assays to measure the immune responses, and this is the overview of what happened in those trials, going from the vaccine design in early January 2020, the first human trial in April, and then moving into late stage trials in the UK by May, and then moving into trials in other countries. Oxford sponsored trials in Brazil, South Africa, and then in Kenya. This is because for a vaccine that we need to use around the world, it's extremely important to have data on how that vaccine behaves in different populations around the world. We can't just rely on data from one country. We have to look at it in different geographies, different ethnic populations, and that was the reason for starting up these other trials. We partnered with AstraZeneca in April. They took on the manufacturing and very early on started large-scale manufacturing in many different sites so that there would be vaccine available for the eventual supply. They also sponsored trials themselves in the US, in Japan, and in Russia. Serum Institute did a trial in India, and by December of 2020, we had the authorization for supply in the UK. So I can skip that one. The efficacy data that came through in November were from the clinical trials run by Oxford, sponsored by Oxford. And of the 4 trials, initially we only had 2 of them included. That was a large-scale trial in the UK and the large-scale trial in Brazil, because at that point the trials, the phase 1 trial in the UK and the phase 2 trial in South Africa hadn't had enough cases in the control group to be included in the analysis. We then added more data from those trials in subsequently. But initially what we were looking for is symptomatic PCR positive COVID-19 with one of those symptoms that are named here in people who had been seronegative when vaccinated, had never had a PCR positive result, and had more than 15 days follow-up after the second dose. We got UK approval in December. And the policy set by the JCVI, it was to use 2 doses of the vaccine 3 months apart. And we'd shown in our clinical trials that the antibody response after the second dose is higher when you have a longer interval between the 2 doses. We also saw very similar results in older people and those with comorbidities. We then were able to update this primary analysis, bringing in more cases, and then this was published in February, but it supported the use of the vaccine. It simply added to the data that we already had. We then moved on to vaccine effectiveness data, which was how the vaccine works in the real world. And we've seen that in England, in people over the age of 70 who'd received 1 dose from 28 days, there was at least 60% protection against symptomatic PCR positive disease. And in the over 80s, hospitalization was reduced by 80%. This is really exciting data to have in people over 70 and people over 80. The vaccine is working extremely well. And despite the different results coming out of clinical efficacy trials, which were run separately and therefore should not be compared, we're actually seeing very, very similar results from the Pfizer vaccine and the AstraZeneca vaccine in these real world effectiveness data, and also seeing that transmission is cut after even a single dose of the vaccine. Now, I just want to spend a moment on the Vax-Hub contributions to this project, because that was really important. In 2020, we had to design the vaccine, order the synthetic DNA we made, start making the vaccine, generate the first stock of vaccine, that's the starting material, characterize that, get it manufactured into clinical trials, and then transfer on the manufacturing process to AstraZeneca and all of their sub-licensees who are now manufacturing the vaccine. And the flexible funding that we had for vaccine manufacturing through Vax-Hub was what allowed us to start this process. The work we've been doing on development of next generation sequencing methods for rapid characterization of the starting material was absolutely critical to our ability to generate this starting material rapidly. In previous years, it used to take us anything up to 9 months, maybe even a year, to be certain of what we had in our starting material. And now we can do it in weeks because of the methods that we've developed through the Vax-Hub funding, working in our own manufacturing facility. The process development for large-scale vaccine manufacturing that was transferred to AstraZeneca was also funded through the Vax-Hub. So without Vax-Hub, we wouldn't have been able to start this project and move it nearly as quickly as we did. So now the vaccine is in use in 100 countries to date. Approvals, in more than 100 countries. It's in use in 168 countries. More than 500 million doses have been released. Around 80 million doses delivered to COVAX, and that's 96% of the total COVAX supply. Obviously, this is a vaccine that we wanted to be used widely around the world. The storage temperature means it's much easier to distribute than other vaccines, and it is being widely used around the world. And to do this, AstraZeneca have built many, many regional supply chains to make that happen. Now, what I haven't told you about in this short presentation is what hasn't been funded in the past. And for 20 years, we have struggled to raise any funding for the development of vaccine technologies. We can get funding for development of a vaccine against a specific disease, but nobody ever wanted to fund technology development, which is absolutely critical to our ability to respond to outbreaks and pandemics. We weren't funded to accelerate the initial generation of the starting material. We had plans in place to do that, to make that happen more quickly, but we didn't get the funding to do that. So we had to start from what we had done, what we knew how to do, what we thought might go better, and make our best efforts to go as quickly as we could. And that's how we were able to get into clinical trials by the 23rd of April, drawing on what we knew about the technology, but feeling at all times very hampered because we hadn't been able to do all of the work that we would have liked to have done and planned to do to make this go even faster. Had we been able to start our clinical trials earlier, it would have been very helpful. So I'm going to leave it there and just give some acknowledgements to the many, many different groups of people who have contributed to this major effort, and particularly the clinical trial volunteers, because without people coming forward to volunteer to take part in clinical trials, we wouldn't have any vaccines. So their participation is absolutely essential. I'll finish there. Thank you.
Thank you, Sarah. So I would move straight on to the Q&A. So if all the speakers from this morning, Sarah, Robin, and Steve, if you'd like to switch on your videos, please. So in this Q&A, I'm going to be supported by Marta Mauri. She's a Vax-Hub early career researcher, and she's going to pose some questions by the audience. I'm going to start with a question for Steve. Which links a little bit to some of the points you've made about the work of the Vaccine Taskforce and what you have planned in the next few years. But we have heard also from Robin and Sarah some of the challenges that they encountered. So my question would be, what really are the priorities the next 5 years in the UK to improve the vaccine manufacturing ecosystem, and maybe linking here to one of the points made by the audience. In particular, that is interesting knowing what are we going to do about variants more generally in the UK?
I think we could take a lot of time answering this question, and I'm just going to give a couple of high level, what I think will make a big difference. I think there isn't enough big player vaccine development action in this country at the moment. So if you look at where the major hubs are, where the major vaccine suppliers of the world are doing their development, it isn't in the UK. And I think that we need to attract that kind of player to the UK. That way we will be at the forefront of technology development of vaccines that are commercially likely to be developed over the next 5-10 years. I think in parallel with that, there has to be a much, I think the way Sarah finished actually struck a chord with me about some of the things that didn't go right in the last 15 months. And I think some of the stuff that Robin's also talked about on technology and how we deploy vaccines is important for us to make sure we've got joined up ways of making sure that our infrastructure uses the investments we've made. And we've got some fantastic institutions. We've got VMIC, CPI, CGMIC, all of these players now in manufacturability coming forward. That needs joining up and a plan putting together for that 5-year period. It's totally different to the variant question. I think I'm going to let Sarah and Robin answer the variant question, because I think I could talk again about that, but actually Sarah could perhaps talk about what she's doing, and that would be a better answer, because that's with government support too.
Okay, so just very quickly then, we have a preprint which came out, I think, yesterday, just showing a preclinical study of giving a variant vaccine either alone or after 1 or 2 doses of the original vaccine. And we see that the variant vaccine induces an immune response which is stronger to the variant, which is the beta variant, than it is to the original vaccine, and vice versa with the original vaccine, but there is some cross-reactivity with either. And then if you've had 1 or 2 doses of the original vaccine and then give a dose of the variant vaccine, you increase the immune response both to the original virus and to the variant virus. In general, the strong immune responses are cross-reactive to many of the variants. So the focus is more on getting a very strong response than a very specific response. This is going to go into clinical trials imminently. But what we don't want to do is end up trying to make multiple different variants of the vaccine and work out how to implement them. The focus is on getting a very strong immune response, which will protect well against all of the different variants.
Thank you, Sarah. May I ask Robin, I know you have typed the answer, but as can be only seen by the person who was asked the question. So, and I think this, your answer is probably going to be interesting for our audience.
So we've also been working on variants. We're not as ahead as Sarah is because her technology has already been approved and licensed. So, we're looking very much more to the future and whether you might need combinations. And I suppose one of the things that our technology offers is the ability to have, 4 or 5 variants in the same vaccine dose. And we are talking to potential partners to fund that. Although we're not currently in the phase of moving it into the clinic. We can move it into the clinic, but it's something that's there should somebody want to call on us to move forward. And I just wanted to pick up on one of the things that Steve said is, we've highlighted 2 technologies, the adenovirus technology, the RNA technology, but actually the hubs are covering a wide range of technologies, and that linkage, that coordination, I think the 2 hubs really started a very good job to link all the different key players together. But that's the beginning of the journey. I think we've created a basis, but we need to look to the future to make sure we build on what we've learned.
Certainly. There are some questions, there's quite a lot of questions from the audience, so we needed to sort of select from. So Marta, what is your, do you have any questions from the audience you'd like to ask our speakers?
Sure. So first of all, thanks for asking so many questions. Some of them have been addressed already, and I'll try to pose the more generic one to the panel. So one was about the variants, which has been addressed. And the second interesting one is whether you are both considering developing a nasal route of administration for your vaccine candidates that would protect from transmission and not only from hospitalization or severity of the symptoms.
So we already know that the intramuscular injection can have an effect on transmission and asymptomatic infection. All of that data has been published. It's less effective against transmission and asymptomatic infection than it is against symptomatic disease and then severe disease. And that's to be expected. But with the Oxford AstraZeneca vaccine, there is currently an intranasal vaccine trial being run. The difficulty is going to be going from the first studies of that vaccine to demonstrating efficacy of the vaccine. It's not possible to measure immune responses and compare them with the immune responses you get from intramuscular injection and assume that the outcome will be the same. So it is necessary to then set up a further efficacy trial, which is quite a large piece of work to be done.
Thank you, Sarah.
So we're also moving into an intranasal study with RNA. That's a really very novel approach. So nobody knows whether it will work. And I suspect that we will need to do quite a lot of optimization on the delivery for RNA technology. So that's something more likely for the future. But obviously, it would be much more amenable for global distribution if we could get that to work.
Thank you. Then a second question that may concern several people is more about the mRNA or self-amplifying RNA vaccine. So actually I put together 2 questions. One is, what is the half-life of these RNAs? And the second one is the concern on whether they could affect the human genome by inserting themselves, for instance. So could you please elaborate, Robin?
So first of all, I'll take the second question first, which it can't change your genome. It can't alter your chromosome. So this is not genetic modification. And I think that's one of the advantages of RNA. RNA typically is quite labile. It stays in the cytoplasm. Now, there are differences between mRNA and self-amplifying RNA. mRNA is expressed very quickly and disappears within a matter of days. Self-amplifying RNA actually is expressed and peaks at about 7 days and can go out for up to 3 weeks. Now, we're interested in that not just for vaccines, which may mature an antibody response, but it could be very important in areas like protein replacement therapy, where you could have an RNA dose once a month, and that's something that couldn't be realized by conventional mRNA technology.
Thank you very much. And maybe I have a third question for all speakers, which is whether we should consider a non-for-profit vaccines or patent-free vaccine. What is your opinion on the topic?
So I can start off. I mean, you've heard from Sarah that AstraZeneca are doing their vaccine royalty-free during the pandemic, and I think that's a fantastic stance, and it would be lovely if the other vaccine manufacturers embraced the same philosophy. Personally, I think that although it sounds very attractive to say waive patents, I don't think it actually gets you to the end game faster, because we know that it's not just patents, it's technology transfer, and the hubs have been looking at technology transfer. That takes time. If you actually partner with somebody like AstraZeneca, you're then covered by their authorization. If you're making a biosimilar, you would have to do a large efficacy trial that would take at least another year. And so it would be a much slower approach to getting vaccines rolled out globally.
I agree with Robin, but I'd also like to add that simply waiving patents and allowing anybody who wants to try to make the same vaccine is going to be really counterproductive. We are all manufacturers are constrained by supply chain issues, and we need all of the supplies that need to be used to make vaccines to go to manufacturing sites that have the technology up and running. AstraZeneca has more than 20 of them. Other manufacturers are looking to expand, where the technology not only to manufacture the vaccine, but to test the vaccine, to do all the necessary quality control and release it in a way that's approvable by the regulators. That's where those resources have to go to. And by making the IP freely available, that doesn't mean that you can make the same vaccine.
And if I could throw my hat in here as also just say that I think one of the other pieces that just what Sarah just said is really important. As you look forward 10 years, what Robin was talking about having a very portable vaccine manufacturing facility is very different. But again, it's not waiving IP, because you'd still need to have people who are trained and able to go with that and operate it and then be able to release a vaccine. It's one of the things I think this crisis has probably taught lots of people in the business is how important the release of the vaccine is at the end of the process. It's not just the authorization, it's the ability to be able to say that vaccine is now usable by the local population. And that's really critical in any of this that we do going forward.
Perfect. Thank you very much. And I think we may have time for one more question, or should I conclude here, Marta?
Yeah, in the interest of time, I'd like to thank the speakers. I know there are lots of interesting questions in the chat. If I can remind people to use the Q&A chat. I know this is confusing. Zoom has a chat and then the similar icon for a Q&A chat. If you could use the Q&A, that would support us in managing the question. And maybe some of the speakers can also view the question and answer, not live, but answer in the Q&A chat in case there is no time to handle those live. So thank you to all the speakers, and I'm going to pass on. Thank you, Marta, and I'm going to pass on to Ben for the hub introduction.